In this video, we will discuss about results of HiFi Cas9 KRAS mutant target system on different cancer cell lines. So in the previous video, we have discussed about the development of this HiFi Cas9 system that can target the mutant KRAS but not the wild type KRAS. So from the cadarnets that they have selected in the previous experiment that we have discussed previously, those cadarnets will be used on different cancer cell lines and at different uh, models such as 2D culture model and 3D culture model to check its efficiencies. So first they use this cell lines that I mentioned here, H358, H1792, A427, SKLU1, H838. Among that, the first two cell lines has these KRAS G12C mutations, and next two cell lines has the KRAS G12D mutation, and the last one has no mutations. And what they are able to see when they culture it with this RNP that contains the KRAS mutant, it contains the guide to target the KRAS mutant, and here the ratio is shown compared to the control. And what they are able to see is that in the case where these KRAS mutants are present, they are able to see a significant decrease compared to the case where the wild type KRAS is present. And thereafter they did their studies on 2D culture and 3D culture model also, and they choose the 3D culture model because the 2D culture model does not replicate the in vivo results. So when they did it for the 2D culture model, they are able to see that for H358 they are able to see 50% reductions in the cell viability compared to the 3D model case where they see 70% reductions, whereas for A427 they see negligible reductions in the 2D culture, but they see 25% reductions, around 25% reductions in the 3D cultures. And for the control where wild type KRAS is present, they do not see any significant difference. So this indicates that this further substantiates their previous perceptions that 3D culture model gives a more better in vivo results. However, they are not shown here so far the results of the in vivo mice, but since they see the high transductions, they are expecting that this results is more closely aligned with the in vivo results. We will see in future experiments how their in vivo results comes out. So far, once they validated that the KRAS mutations goes down in different cell lines, they wanted to see whether it will also affect the downstream KRAS signaling pathways or KRAS related signaling pathways or not. So here, and they compare it with this RNP HiFi Cas9 system, and simultaneously they also developed the adenovirus associated HiFi Cas9 system, where instead of RNP they use adenovirus to transfect into the tumors, as in most of the therapeutic conditions adenovirus has been regularly used for the therapeutic drugs. So for the KRAS mutations, they are able to see reductions in the H358 mutant of the G12C, and simultaneously they also see reductions in these phosphorylations of the ERK pathways and these PHP70 pathways. And the results are also shown quantitatively in the right hand side of the panel D, where we can see that there is a reductions compared to the wild type in the KRAS and P70 signaling P70 proteins, and also phosphorylated P70 proteins and phosphorylated ERK proteins that can be seen here. And when I did the same studies for A427 for the G12D proteins, they are also able to see it. In this case, they completely see reductions in the KRAS signals and also phosphorylated ERK signals, but you do not see, and also a significant decrease in the phosphorylated P70 signals. And the quantifications on this is shown on the right hand side, where we can see a decrease in this KRAS, phosphorylated P70 and phosphorylated ERK. And when I did the similar studies with the adeno-associated virus HiFi Cas9, they are able to see less reductions in the KRAS and the phosphorylated 70, and also there is small reductions in this phosphorylated ERK. And the reduction in the efficiency of adeno-associated virus HiFi Cas9 is because the efficiencies of the adeno-associated virus HiFi Cas9 is lower than the efficiencies of the RNP HiFi Cas9 system. This is what they point out the reason why the results from the adeno-associated virus HiFi Cas9 is low. So far, this is what they have presented in the figure. We will discuss further results in the subsequent video. If you need to understand more about these results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.